Science is a very broad term. The goal of science is to understand the natural world. Within this goal are many subdisciplines, including understanding forces, which is called physics, understanding matter, which is called chemistry, understanding composition and changes of the earth, which is called geology, and many more. Biology is the study of life. Within biology, there is the study of animals, which is zoology, the study of plants, which is botany, the study of bacteria and fungi, which is microbiology, my personal favorite, the study of insects, which is entomology, and many more. In high school, you likely learned a little about the scientific method. For this class, we will use it to make our own experiment regarding the Madagascar hissing cockroach. We first must make an observation so we can then form a research question. Once we have a question, we are able to form a hypothesis. A hypothesis is a tentative explanation of the observed phenomenon and a prediction of what will happen under experimental conditions. We will then perform the experiment and analyze the results we get. If our results are conclusive, then we can report our findings. However, if the results are not conclusive, then we perform the experiment again, or we reconfigure the hypothesis. Let's look at an example of experimental design. You might notice that your rose garden grows larger after a rain compared to when you water it with a water hose. You are curious enough to want to find out if this is true, so you research the optimal growth requirements for roses in a gardening book, and you look on the Environmental Protection Agency website to see if any air or groundwater pollution exists in your area. You find a particular contaminant is on the high side of normal with regard to human health in the aquifer from which you draw your well. This gives you a hunch the contaminant may be affecting your roses. You hypothesize rose bushes receiving well water will not grow as much as rose bushes receiving rainwater, because the well water has slightly elevated levels of contaminant X. The next step is to test it with an experiment. You decide to water some rose bushes with only rainwater and some with only well water. This seems pretty straightforward, but there are so many other considerations when growing roses. From your gardening book and personal experience, you know that the temperature, amount of water, amount of sunlight, use of fertilizer, and many other factors contribute to successful rose growth. To increase the likelihood you get results from what you are trying to test and not something else, you must control all those other factors. In this case, you want to make sure only contamination of the water source affects the growth of the roses. So, you will need to make sure all rose bushes are treated exactly the same with respect to all of these other factors. These are called controlled variables and are defined as all variables except the variable being tested that could have an effect on the outcome of the experiment. The variable being tested is referred to as the experimental variable. In this case, it is the source of water. Groups that are treated differently in an experiment are called treatments. If you are applying an experimental variable, it is called an experimental treatment. In the example of the rose garden, plants receiving well water and plants receiving rainwater are the two experimental treatments. Any experiment will have one or more experimental treatments. Some experiments will have a control treatment from which the experimental variable is withheld to serve as a baseline. For example, if you were testing fertilizer on your roses, a control treatment with no fertilizer would be used for comparison with the experimental treatments with fertilizer. Not all experiments have a control treatment because of the nature of the experiment, but one should be included whenever possible. Comparison type experiments usually do not have a control treatment. In the comparison of water sources for roses, eliminating both rainwater and well water could mean no water at all and would certainly have an effect on the growth of the roses. But it would not be due to a water contaminant. Therefore, a control treatment would not be applicable in this case. Sometimes random factors affect your experiment that cannot be predicted. Some seeds are going to grow larger rose bushes than others regardless of the type of water used. If you are only comparing one rose bush for each water source, the random factors may lead you to draw an irrelevant conclusion. However, if you have many rose bushes receiving rainwater and many rose bushes receiving well water, you will probably feel more certain about your conclusion. The role of chance and uncontrolled variables are less likely to affect an experiment with many trials. Replication, which is the use of multiple trials of the treatments, is a good way to increase certainty by reducing the effects of random chance. Controlling variables, performing replication, and including a control treatment are just a few elements of experimental design that help increase the likelihood of the results being due to the factor you are testing. Another way of saying this is that you are reducing assumptions in your experiment. An assumption occurs anytime you assume a factor has no bearing on the outcome of your experiment, when it does, in fact, influence your results. For example, if some of your rose bushes are planted in pots while others are planted directly in the ground, you might assume there is no difference as long as you controlled all the variables previously identified. However, there might be effects from the different ways of containing the plants that affect the way they grow. You cannot eliminate 100% of all assumptions, but an experienced scientist knows how to spot the ones likely to lead to enormous findings. You expect manipulation of an experimental variable, also known as the independent variable, to cause a difference in the result, or dependent variable. A good way to remember the difference between independent and dependent variables is that the dependent variable, or the result, depends on the independent variable, or the experimental variable. In the example of the growth of the rose bushes depends on the source of water. The source of water does not depend on the growth of rose bushes. Rosebush growth can be measured several ways, such as the height of the bush, the number of flowers, or the number of leaves. The dependent variable must be specified precisely, and it must reflect any potential influence by the experimental variable. From earlier observations, perhaps rosebush height was the effect you initially noticed, so you could make the height the dependent variable. We are now going to jump ahead into Chapter 3. So, if you want to turn into your lab manual to page 16, you can follow along from there. Looking back on the scientific method, we have now made an observation, asked a question, formed a hypothesis, and performed an experiment using these rose bushes. So, we are now at the point where we need to analyze results. In this case, we're going to look at the length of Madagascar hissing cockroaches. We're going to measure the difference between males and females. In your head, I want you to go ahead and come up with your own hypothesis. Which do you think will be longer? Do you think that males will end up being longer or females? We are now going to look at the distribution of size of Madagascar hissing cockroaches. We have 3 females and 3 males for this experiment. X's are female, Y's are male. We use these numbers and then take an average, or find the mean. In this case, the mean of X is 57 and the mean of Y is 54. Looking at this, you might say that the female is larger than the male. But if you look at each individual number, females not only have the largest cockroach, but they also have the smallest. This is considered variance. We also need to look at the overlap. So, both males and females have fairly similar sizes if you look at it. Males seem to be more consistent with their size, at least of the ones that were chosen, but they also have a great deal of overlap when it comes to the same size as the females. This is a standard bell curve showing normal distribution. Some bell curves are short and wide, indicating wide variation within the data set, and other bell curves are tall and narrow, indicating more individuals falling closer to the mean and little variation. If you put two sets of data on the same graph, both males and females in this case, you could compare the two treatments visually. We're now going to look at a couple of examples. In this situation, you find the mean length of males and females is the same or nearly the same. However, the range in one sex varies greatly, while the other sex varies to a lesser degree. You have the same mean, but a different variance. This leads to a shorter, wider bell curve on top of a tall, narrower bell curve. In this situation, the variance from the mean is the same or similar, but the means are quite different. The first situation, the overlap is tremendous, while in the second situation, there is almost no overlap at all. Other possibilities are different means as well as different variance. This also happens often. We're now going to look at something called the student's t-test. The student's t-test is used to determine if the means differ enough and the variance and overlap are low enough to accept the hypothesis. Shown is the student's t-test formula. X represents the data for one data set. Y represents the data for another data set. X bar and Y bar represent the respective data set means. S represents the standard deviation. The subscript denotes the data set. N represents the number in the sample. The subscript also denotes the data set. And you take the absolute value of the formula. The negative sign is dropped if the answer is negative. Before you can perform a t-test, you must first perform a standard deviation. This is to account for how far each value deviates from the mean. But you can also get a lot of variance by if you have a lot of data. So this is adjusted with the sample size in the denominator. Y is the variance squared. If it was not squared, then it would equal 0, and we don't want that. Y is 1 subtracted from the sample size to ensure you have a replication. So I'm going to put portions of the example up for you to look at. We will do more in class later, but this gives you a general idea of what you're doing and what the t-test is. So you take S to equal the square root, and you take all of your data set samples. This eventually comes out to 6.56. Now that we have our standard deviation, we are able to plug those numbers into our t-test formula. Shown below are now the standard deviation plugged into the formula as well as the rest of the variables. This gives us a t-value of 0.73. In our student t-test formula, we have the numerator, which compares means, the denominator, which accounts for sample size, accounts for variability, and outliers have less effect if sample is large. Our final formula that we are going to use is called degrees of freedom. This formula is fairly simple and is used to indicate the number of independent variables that can vary in an analysis without breaking any constraints. This one is simple enough. You simply take the number of variables in X, which in this case was 3, minus 1, plus the number of variables in Y, which was also 3, minus 1. Now that we have calculated all of our formulas, we are able to look at the t-distribution table. This can be found in the back of your book. This table shows the degrees of freedom, highlighted in green. Once you have your degrees of freedom, you're able to look at only this row. So in this case, we will only be looking at the row of 4. All of the numbers that are in purple are the t-values. So we will take our t-value of 0.73 and find it on our table. You are able to see that our t-value is less than any of the numbers found on the table. This leads us to look at our p-value. The p-value is the probability the results are just due to random chance. In this case, our p-value is 0.25, or 25%. While in many cases this may seem like a pretty decent result, 25% is considered not very good. We typically in science want to cut it off at about 5%. Otherwise, it is more likely due to random chance. The bigger the t yields a smaller p. A bigger n, or number sample size, yields to a smaller p. If our p-value is less than 0.05, it supports the hypothesis. However, if it is greater than 0.05 or 5%, it does not support our hypothesis. Smaller p means less probability results are due to chance. When analyzing your data, you need to consider whether your hypothesis has one tail or two. One tail is when your hypothesis predicts directionality, like you predict male MHCs will be longer on average than females. This typically also falls under bigger, smaller, more, less, higher, lower. Or you could have two tails when your hypothesis predicts only a difference. For example, you predict there will be a difference on average between male and female Madagascar hissing cockroaches, but you don't know which will be longer. Because a two-tailed hypothesis is less specific, the t-test is more stringent. The t does not change, but the p does. On the t-distribution chart, the p-value shifts one column to be 0.10.